Okay, sounds great. So I'll keep it quick to the point because I think we are already 45 minutes behind schedule. Well, this is a new molecule, Enmetazobatam. I'm not sure whether you're aware of it. This is the first ever India-invented molecule which has been approved by US FDA. And the credit goes to Orchid Pharma. And I'm proud to be that lead co-inventor, the biologist who led this discovery. Yeah, I'll skip all this, the history of Orchid. I'll not even go into what is UTI. You all know the prevalence. Okay, you're looking at, on the left side, Tazobactam molecule. All right? On the right, you look at it, it's almost same as Tazobactam. That brings in a differentiator, especially to tell you that the safety profile of this molecule has to be good because it's the same class of molecule. It's a beta-lactam class. So when you add a methyl group on the top, you can see there in the red I put a methyl group, and we have a negative charge on the carboxyl molecule at the bottom. And if you look at cefepime on the left, even it has got a same kind of a positive and a negative molecule, nitrogen at the positivity and a O negative. The reason I'm mentioning a bit of chemistry here is when such a nature comes into a molecule, it becomes zwitterionic. If a molecule is zwitterionic, it can penetrate the cell very well. So if it penetrates the cell very well, then the site of action is well taken care of, right? So if you have a beta-lactam and a beta-lactamase inhibitor with similar properties, the efficacy significantly improves. So instead of going to a carbapenem right away, you don't have to do that anymore because you have cefepime and enmetazobatam which can take care of those infections, especially in urinary tract, pyelonephritis, and especially complicated UTIs. Pyelonephritis, you have HAP, WAP, and also bacteremia ensuing because of this due to the indications. This is a structural chemistry, again very foreign to you all, most of the clinicians. These are the two structures of the beta-lactamase enzyme. If you see Enmetazobatam, it binds quite strongly and in multiple sites within the enzyme. So what happens is why we give the dosage you must have been wondering why I give a different combination of piperacillin and tazobactam and why it is not equal concentration. Now cefepime and tazobactam is given in equal concentrations, 2 gram with 2 gram. But here you don't have to do that. It's 2 gram with 0.5 grams. The reason is the way it binds into the socket, it stays there for a longer period of time. So the number of cycles which the enzyme circulates, the molecule circulates within the enzyme is very low. So when you have low turnover number, you need less concentration of the drug. Okay? That's why this was, if you see the numbers at the bottom, Enmetazobatam you got about 100 units, whereas Tazobactam is 500. This takes a while to move. I'll skip this. And this is essentially to show you how the molecule behaves compared to other molecules which are existing in the market. You have got everything which is available, which are in purple, and orange is the new molecule, and you'll see the efficacy is 98.9% potent molecule. So you don't have to reach your hands to carbapenems. And this study is from more than 7,000 strains tested, and again you will see similar kind of data. I will take you to this question every clinician will ask. Okay, you did the discovery, but what is the current status in India? Does it work on Indian strains? This study was done in Indian strains, about 300 strains, and you will see the top one is, these are all E. coli sensitive strains, and you've got ESBLs, AMP-C producers, and Enterobacter, and you will notice that cefepime Enmetazobatam shows nearly 100% of its activity compared to other combinations which are existing in the market. This is with the current strains which is done in just the month of August. And you'll also see again Klebsiella, it shows a similar picture, right? So this is a very promising molecule. That's the reason I'm telling you why this is a better choice. Not because it's my baby because it's got good characteristics. All of us love our own children. Now, this data is published data from 2 different studies. You will notice meropenem efficacy, potency compared to cefepime and enmetazobatam. In fact, you will see the potency of enmetazobatam is much better, combination 0.25 and 0.5 at various concentration compared to meropenem alone. So these are the data which has brought this molecule to be positioned as a carbapenem sparing molecule as a first line of treatment. Okay, these are the main characteristics of the molecule, which I want to, as a snapshot. The dose is 2 grams cefepime with 0.5 grams of enmetazobatam, and it's used for the indications CUTI with HAP, VAP, and also the activities against ESBL producers, and also Pseudomonas and aeruginosa, and also Acinetobacter OXA48 producers, and TEM, SHV, CTXMN, against all these molecules it works. So safety and tolerability, it's going to be similar to that of any beta-lactam group of molecules. So you need to bear in mind the hypersensitivity reactions are going to be similar to any beta-lactam class. And positioning globally, it is to be given, in India it is still phase 4 has to be completed, but US FDA, UK MHRA, EMA has approved it for marketing already against all the indications I mentioned. In India, it just got launched 3 days ago on 1st of October. All right? You will see the name as Orblicef. That's the molecule. Orblicef is the molecule which is being, it is the brand for, OR is for Orchid, BLI is beta-lactamase inhibitor, and CEF is cefepime. That's the branding of Orchid Pharma for this molecule. So I encourage all of you to look at this molecule. You will see it in your clinics already, and I think it's going to increase the numbers. Thank you very much.
Thank you, Maneesh, for, first of all, congratulations to you for this new discovery. And second, to sticking to the time, saving us some time for the next session. This is a very, very important discovery considering the scenario. We all know for last few decades, we haven't got any new antibiotics. Pipeline was almost dry. And we have something coming up which is very effective, which can spare use of carbapenems. But one word of caution to you all, since now it is available in the country as well, do not misuse this.
Absolutely.
Because resistance, although, at present they don't foresee resistance developing to this molecule. I remember when I was MD student, and at that time fluoroquinolones were launched, and it was said resistance cannot develop to fluoroquinolones. Now we see all fluoroquinolones having resistance being developed to that. So let us not misuse it. Use it sparingly where actually it is needed, so that we can prolong the effectiveness of this new molecule.
But I would suggest one small thing there. Yeah. It should be used to spare the carbapenems, because we need carbapenems for difficult to treat infections. If that becomes the first line, then we don't have anything else to go for. So that's the only caution I would say, please spare the carbapenems so that we save them. Yeah. And the beauty of this whole invention is, this is the first invented in India molecule, which the world has taken it up. So it's a pride moment for a country as well.
Congratulate him, give it a huge round of applause.
Thank you.